Hello, and welcome to another episode of Research Aromatica. Today, I am very pleased to introduce you to today's speaker, Joyce Kelly do Rosario da Silva. She's going to present to us today the chemical profile and biological activities of essential oils from the Amazon. Joyce holds a PhD in organic chemistry from the University of Alabama at Huntsville. She is currently an associate professor of the Faculty of Biotechnology of the Institute of Biological Sciences and graduate program in biotechnology at UFPA. She is also an affiliate member of the Brazilian Academy of Sciences and has a productivity scholarship of the National Council for Scientific and Technological Development. In addition, she was the first woman in the Amazon region to be laureated with the prize for Women in Science by UNESCO, L'Oreal, and the Brazilian Academy of Sciences. In the last 10 years, Joyce has published over 80 papers in the field of chemistry, of natural products, especially essential oils and aromas. Her research interests involve the study of chemical composition, bioprospecting, variation of metabolites during plant pathogen interactions, and development of new products from essential oils and the molecular biology of plants. That's an impressive bio, Joyce. It's wonderful to have you here with us today, live streaming in from Brazil. Thank you for being here. And so we can get your slides started and get the presentation going.
Okay. My presentation title is Chemical Profile and Biological Activities of Essential Oils from the Amazon. First of all, I will start my presentation with this question: why it is important to know about the aromatic flora from the Amazon? The territorial extension of Brazil is divided into 6 natural biomes. Each biome has a specific fauna and flora and the climatic conditions. The Brazilian Amazon, represented by the green color area in this map, covered 9 states, which 7 belong to the northern region. The Amazon territory corresponds to 40% of Brazilian area. In this biome, the most biological diversity of the world is observed. In this sense, the Amazon region represents for us a good place to prospect for new bioactivity molecules and new raw materials. Aromatic plants in the Amazon region are used as spices in the preparation of traditional dishes, and some of them are used as flavoring agents or like fragrance, and few species are explored to big companies to production of perfumes. However, the aromatic plants are mostly used as infusions and commercialized in an open-air market due to their medicinal applications as anti-inflammatory, analgesic, anti-diabetic, and in the treatment of many diseases. In this sense, we promoted the systematic studies of aromatic plants from the Amazon. Our systematic study is based in 4 main factors. The number 1, the species are under permanent environmental pressure due to the human action in the exploitation of the forest resource, which is still very predatory in the region. The number 2, the extinction rate of this aromatic species is very high and is so difficult for the recollection for a plant from the same place. Number 3, the establishment of inventories of the Amazon aromatic flora is necessary to increase the source of new raw materials. Number 4, the extensive area of the Amazon region and the absence of scientific and technology knowledge about the plants with economic potential motivate us on this journey. In addition, the systematic study can avoid a repetition of the situation that happened with the rosewood in the past. This plant is very known in the Amazon region and produced an essential oil rich in the monoterpene linalool with amounts around 92%. This essential oil has an economic importance to the fragrance industry and a high exportation. Unfortunately, due to the over-harvest in the past, the rosewood is now listed as a species with extinction risk. Our database of aromatic plants from Amazon are based in 5 main experimental steps. The first one is the botanical identification. Number 2, essential oil extraction. Number 3, chemical composition analysis by CGMS. Eventually, some samples are submitted to column chromatography for the isolation of its major compounds, and then submitted to biological assays to evaluate the pharmacology potential or cosmetic applications, among others. And the last, we have the insertion of this information into the database. In this picture, I'm showing some collection sites in the Amazon, which we have collected plants, aromatic plants, in the last years. Here we have Jalapão, Carajás, Chapada das Mesas, Quexuanã, and Serra das Andorinhas. The main information present in our database consists in the scientific name and the common name for each plant, and the plant tissue which was extracted the essential oil. And we have information on oil yields and the chemical composition and chromatogram obtained by CGMS. And we have also information about the habitat and geographical distribution and agronomic characteristics and traditional uses for each plant. Based on our database, the most representative aromatic species in the Amazon belong to these botanical families. The first one is Peperoma, followed by Martaceae, Asteraceae, Lamiaceae, Annonaceae, and Lauraceae. These botanical families showed a chemosystematic relationship. For example, in the species from Verbenaceae family, the monoterpenes showed a higher distribution. On the other hand, sesquiterpenes are the main compounds identified in Martaceae family. In addition, phenylpropanoids are the most representative compounds class identified in the Piperaceae species. Likewise, we also found a relationship between biological applications and the chemical composition of the essential oils. The oils rich in the sesquiterpenes showed a high cytotoxic activity against different cancer cell lines. Essential oils rich in phenylpropanoids displayed a high antifungal potential against phytopathogens and human pathogens. Additionally, essential oils rich in monoterpenes and phenylpropanoids showed a good radical scavengers and promoted an inhibition of lipid oxidation. Finally, the oils rich in the terpenoids and phenylpropanoids showed a good pharmacological potential as anti-inflammatory and analgesic. Now, I will show some results obtained to essential oils with anti-cancer potential. The cytotox effect of the samples was determined by MTT assay. This assay is based on the measuring of the cell availability after the exposure to the essential oils. The oils were tested against different cancer cell lines as breast cancer, colon cancer, melanoma, and gastric cancer. In this study, we evaluated the cytotox effect of this plant, Piper aequale. This plant was collected in the Carajás forest, and the essential oil was obtained by hydrodistillation and presented as the major compounds delta-ilimine and beta-pinene and alpha-pinene. This sample showed a high cytotoxic activity against gastric cancer cell lines with an inhibitory concentration of 1.54 microgram per ml. When we analyzed the cell death mechanism, we observed that this essential oil promoted the cell death by apoptosis at concentration from 0.75 to 3.0 microgram per ml. In this another study, we evaluate the cytotoxic activity of Eugenia Uniflora. This plant is very known in the Amazon region and has an economic value due to the edible fruits. In addition, the leaf and the bark tea has medicinal properties and used by local communities in traditional medicine in the Amazon. The oils were extracted by the leaves, from the leaves, and the CGMS analysis showed a high concentration of oxygenated sesquiterpenes and sesquiterpene hydrocarbons as predominant in the samples. We identified four different chemical profiles. As we can see in this graphical, the principal component analysis, a multivariate statistical analysis, classify the oils into 4 chemotypes. Some of them were rich in oxygenated sesquiterpenes, and others presented a high concentration of sesquiterpene hydrocarbons as caryophyllene oxide, caryophyllene, germacrin B, and quercetin. The chemotype number 4, rich in quercetin, was submitted to chromatography column for the isolation of its major compounds. The column was evaluated with a mix of petroleum water and N-hexane in a rate of 1:4 and monitored by thin layer chromatography for the isolation of cusurin. In this table, we have the results obtained to cytotoxic activity of Eugenia Uniflora essential oils on different cancer cell lines. We tested the oils against colon, gastric, melanoma, cancer cell lines, and we have good results to the sample number 4, which is rich in cusurin, and cusurin isolated showed the same effect. In this sense, the anti-cancer activity was attributed to the high concentration of this sesquiterpene. When we evaluated the cell death mechanism of the cusurin, we also observed that the cusurin induced the cell death by apoptosis at concentration from 2.5 and to 10 micromole. In continuing talk about cytotoxic activity, in this study we evaluated cytotoxic activity of 7 Martensi specimens collected in Pará states during the dry season. The essential oils were obtained by hydrodistillation and showed a high concentration of terpenoids such as sesquiterpenes and monoterpenes as caryophyllene, germacrin, and germacrin B, and epi-bisabolol, and alpha-pinene. In this table, we can see the cytotoxic activity of the Myrtaceae species against different cell lines. All samples presented a high cytotoxic activity, except against the cancer colon lines, which present good activity only to 2 oils. The oils obtained from Eugenia Patrice and Mircea splendens. Now, I show some results obtained to essential oils with antifungal activity. The antifungal activity of the essential oils was evaluated followed by this protocol. The oils were dissolved in dimethyl sulfoxide in a proportion of 50-50%, and then the samples were incorporated to the culture medium at concentrations from 0.5 to 5.0 microliter per ml, and the disc containing the fungal mycelia was inoculated. The plates were incubated under different conditions of light and temperature, and at 10 days post-inoculation, the mycelial growth was calculated. In this study, we evaluated the antifungal and chemical composition of 2 different chemotypes of cinnamon. These 2 samples were collected in Maranhão state and present a different chemical profile. The first one presented a high concentration of eugenol with amounts of 90%, while the sample number 2 showed a high concentration of benzyl benzoate. These two samples were submitted to antifungal assay against this fungal, which causes the disease anthracnose in bananas. The sample number 1 and its major compound, eugenol, showed a total inhibition of the mycelial growth. However, when we evaluate the oil rich in benzyl benzoate, the inhibition was only around 80%. In addition, when we evaluate the antifungal activity of benzyl benzoate isolated, we have an inhibition only of 55%. In this other study, we evaluate the antifungal activity of the Piper de varicato essential oils against Fusarium infection in black pepper. The sample was collected in Belém, and the essential oil was extracted by hydrodistillation and showed as main compounds methyl eugenol and eugenol. The antifungal activity of the oil of Piper de varicato and the pure compounds was evaluated by the SEMI protocol, and eugenol isolated showed a total inhibition of mycelial growth at concentration of 0.75 microgram per ml. Based on the good effects observed to the major compounds, we evaluated the synergistic effect of these major compounds at different combinations. All samples were tested at concentration of 0.75, and eugenol isolated showed a total inhibition of mycelial growth, while methyl eugenol, when I was isolated, showed inhibition only of 8%, while when we have eugenol and methyl eugenol in a combination of a concentration rate of 4:1, we have this effect potentialized by the presence of eugenol. In this study, we concluded that the eugenol is the main compound responsible for this activity. Continuing talk about essential oils with antifungal action. In this study, we evaluate the antifungal activity of the essential oils of Piperadunco, which is rich in dilapiol, a phenylpropanoid, and the plant was collected in Belém, and the leaves were submitted to hydrodistillation, and the essential oil rich in dilapiol was submitted to the chromatography column, and then we have a purified fraction rich in dilapiol at concentration over 90%. The samples, the oil and the fraction rich in dilapiol were submitted to antifungal assay based on the microdilution method, and The samples showed a high antifungal activities against all these fungi. They showed a good minimal inhibitory concentration and in the range from 250 to 500 microgram per ml. We considered these samples as a good antifungal agents. Based on our excellent results, we developed a pharmaceutical composition containing nanoparticles of dilapiol for topical administration in the treatment of cutaneous infections. We developed a formulation which promoted the high stability to dilapiol and the best cutaneous permeation. And we recommend this formulation to the treatment of infections caused by the fungus Trichophyron rubrum. Now, I will show an example of essential oil with cosmetic applications. The oil was extracted from this plant, Lipia oreganoides. This plant is aromatic shrub, which grown in savanna areas of North Brazil, and many studies have reported its biological activities. The leaves are used in traditional medicine as antimicrobial, antihypertensive, and anti-inflammatory, and as analgesic and antioxidant. This species presents different chemical profiles. The most frequent chemical profiles are characterized by the presence of thymol, carvacrol, and cineol. In your study, we evaluate the tyrosinase inhibitory activity. Why we checked this activity? Because tyrosinase is an enzyme which participates in the last 3 stages of the melanogenesis. A tyrosinase inhibitor would serve to block the melanin biosynthesis, and a compound which presented this property would be important as a base of formulations used for the treatment of skin blemishes. In your study, we identified 5 different chemical profiles. The first one was rich in monoterpenes and sesquiterpenes as cineol and alpha-terpineol and caryophyllene. And the chemotype number 2 presents a high concentration of thymol, a phenolic compound. And the chemotype number 3 showed a high concentration of the monoterpenes alpha-philandrine and beta-philandrine, and also showed a high concentration of beta-caryophyllene. And the chemotype number 4, we have monoterpenes as alpha-pinene and sabinine and cineol. And the chemotype number 5, we have high amounts of carvacrol, beta-caryophyllene, and nerolidol. These all oils were submitted to the tyrosinase inhibition assay and presented different inhibition rates. For example, the thymol chemotype showed the highest inhibition of the tyrosinase enzyme. The effect observed to this essential oil was similar to the observed to the standard control, the kojic acid. And we conclude in this study that thymol can be used as an agent in pharmaceutical formulations which promote skin blemish. Based on results, we developed a pharmaceutical formulation containing thymol nanoparticles to increase the resistance of thymol to temperature and increase its solubility in water and control the drug release. These papers, we can see all steps of the preparation of this pharmaceutical formulation. Based on the results, as a good essential oil with cosmetic application, we developed a method to measure of the skin permeation of thymol. We observed that thymol displayed an excellent recovery in all skin layers. We evaluate the permeation in the strata corneal and the hair follicle and in the remaining skin, and we observed a recovery to thymol around 90 to 100%. Now I will show some samples with pharmacology potential. The first one is Aniba canelilla, is a Lauraceae species. This species is very known in Amazon region as fake cinnamon, and the barks of these plants are used as tea in traditional medicine, and some other tissues as leaves are used as spices in the preparation of local dishes and fragrances. The essential oil extracted from the bark is dominated by the presence of these nitro compounds, 1,nitro-2-phenylephrine, with amounts of 60 to 90%. The concentration of these compounds in the essential oil, in the Aniba canelilla essential oil, is very high. And in this study, we evaluated the acetylcholinesterase inhibitory activity of the 1,nitro-2-phenylephrine, and we obtained very expressive results because the major compound of the Aniba canelilla essential oil showed activity equivalent to the standard control, the alkaloid physostigmine. And why we are looking for acetylcholinesterase inhibitors? Because these compounds are considered as promising therapeutic agents in the treatment of various neurological disorders such as Alzheimer's diseases and Parkinson's diseases. The samples were tested by the assay biotography. Finally, in this paper, we evaluated the antinociceptive and the anti-inflammatory effects of the Peperoma serpes essential oil. This plant is very popular in the Amazon region, and the common name is carrapatinho, and its leaves are largely used in Brazilian folk medicine to the treatment and the pain and inflammation. We comproved this property to the essential oil, and we found significant effects in the treatment of inflammation and pain. The essential oil obtained by hydrodistillation showed a chemical composition rich in sesquiterpenoids as nerolidol and ledol and alpha-humulin. Finally, I want to introduce our research team. Here we have Dr. Maya and Dr. Eloisa, me, and Dr. Pablo, who composed our research team. And I would like to say thank you for the scientific collaboration of Dr. Setzer from the University of Alabama in Huntsville. And I finished my presentation. Say thank you for having me today, and I leave in an invitation to visit Belém. An invitation to visit Belém, my city.
Thank you so much, Joyce. I definitely want to visit you in Belém. It's an incredible amount of research that a team of 4, 5 scientists are doing. It's extraordinary and inspiring. We do have a couple questions for you. When you were discussing around anti-cancer activities, we had a question around, are you doing any in vivo studies in animals or humans around anti-cancer?
We have some studies in animals, but not yet to humans. But if you have a good result, we wanted to extend this study to clinical assays in humans in the future, maybe.
Yeah, I think Dr. Satyal is complementing your interesting research work. And Dr. Satyal, if you want to expand on, I think you're saying you did some in vivo studies, please feel free to expand on that in the chat. And in the meantime, another question for you is, how many plants are in this database, and how is the database used?
In our database, we have a registration of around 3,000 plants. Wow. But some of them belong to the same species, but we have about 1,700 different species, you know?
Wow, impressive. Yeah. Do you also work in the field collecting the plant material?
Yeah. But
so you do both. You're in the field and the lab.
We stopped in the last year because we have the pandemic, and we stopped to collect field. But we are doing it all the time.
Yeah, and how is the database used, Joyce? Is it used between academics? You did mention developing some pharmaceuticals. Can you expand on that a little bit?
The database can work by this way. If you are looking for essential oil rich in thymol, we can insert the compound name in our database, and you have a list, a special list which presented essential oil rich in thymol, you know. Or we can, and when you have this this information, we can see how many species presented as major compounds, where we can collect it, and which species is good to cultivation, you know, and we can explore this if you can explore it, this species of economic point of view, you know.
Yeah. And as you said in the beginning, some of the predatory collection of plants, it's so important the work your team's doing is cataloging and analyzing these plants from the Amazon, which is a gift to Brazil and certainly the entire planet, all of the plants.
We have some species that presented a very unique composition, chemical composition, but we did not see this plant again because when we back through the same place, this plant was removed to the habitat, and we did not find.
Yeah, it's a reality that we're dealing with.
Yeah.
So important. So important, the work that you're doing. Lucia asks if plant Peperoma is a tree invader. Is it Peperoma?
Yeah.
Is it a tree invader?
Yeah.
It is.
It's a tree invader. Yeah, it is.
Okay.
But we have many species with interesting biological activity in Brazilian Amazon.
Yeah, you bet. Lucas says amazing work, and that very glad I had the chance to be a part of this team under Dr. Joyce's supervision until last year. So you've certainly touched the lives of many students, as well as your colleagues. Yeah. And so thanks for that, Lucas. I think I have asked the main questions to you. And so I want to thank you very much, Joyce, for taking the time to be here with us today to give this talk. Your work is incredibly important, and it's very inspiring to follow you as a woman scientist in chemistry, and to see all the awards and accomplishments that you've made as a woman scientist. It's a pleasure to have been able to host you today. Thank you so much, Joyce. And thank you to our audience.
Thank you.
Great.
Have a good day.
Bye.
Bye.